this sac-like structure is called a sporangium. And consequently, the spores inside are called what, do you think? Sporangiospores. Sporangiospores. A lot of these fungus terms, it's kind of like German, where you just take one word and you add it to another word, you can get a new word. Okay. So the sac-like structure is called a sporangium. And what's the name of the asexual spores inside? Sporangiospores. You nailed it, you guys. Good. Now what we'll do is we'll look at another cartoon of Rhizopus undergoing sexual reproduction. And there's a little cartoon right on the right-hand side. And in addition, on page 3, the cartoon labeled B is a nicer cartoon of Rhizopus undergoing sexual reproduction. So we'll just show kind of the end products. So here's Rhizopus undergoing sexual reproduction. Okay, now we want to set the stage, you guys, for Rhizopus undergoing sexual reproduction. And my understanding is that when the environment starts to deteriorate, for example, if a nutrient started to be used up, maybe a lot of metabolic waste products are accumulating, it will trigger sexual reproduction in Rhizopus. And to me, this makes so much sense. It's like, if the environment is stable and you've succeeded, you've survived, it makes sense to just reproduce asexually, right? Just make copies of yourself. The genes you have made you successful in that environment, right? But if the environment starts to change, this is when you would want to trigger sexual reproduction. Because the environment is changing, you need to generate some genetic diversity in your offspring to hopefully assure some of them will survive in a new environment. So this makes sense to me that sexual reproduction will be triggered when things start to get, bad, start to change. Now, sexual reproduction, like we said, it requires 2 genetically different parents, okay, to be present. And we don't use male and female. Instead, they use pluses and negatives to label the 2 parents. So we'll just say this is the plus strain, and this is the negative strain. And we just want to remember they're genetically different, genetically different. What they do is they communicate using chemical signals called pheromones, just like humans do. Yeah, they send out chemical signals, and it lets one fungus know that a genetically different partner is in the neighborhood. And as a consequence, they will grow these little side branches of their hyphae. And when the hyphae from the 2 parents meet, this is when the fertilization process occurs. The gametes combine genetic information from the positive strain and the negative strain. And it gives rise to a brand new structure, a sexual structure. Now there's 2 names that I would be totally fine if you use either one. Some books you'll see call this new structure the zygosporangium. And other textbooks call it the zygospore. Either name, you guys, would be fine on the lab practical. What we want to recognize is the product of sexual reproduction. Okay, sexual reproduction. Okay, so again, this is what makes it so hard. This is Rhizopus undergoing asexual reproduction. You'll have a slide of Rhizopus undergoing asexual reproduction. You'll see the sporangium with the asexual sporangiospores. You'll have a separate slide of Rhizopus undergoing sexual reproduction. And you'll be identifying, luckily these things are huge. They're really easy to see. These dark brown to black zygospores or zygosporangium, a product of sexual reproduction. So you can see Rhizopus looks really different. Asexual versus sexual. Okay? All right, you guys okay with that? All right, let's see, what else do we have? All right, now down there below the Rhizopus we have Penicillium. And it turns out in nature, and one of your colleagues asked this question yesterday. It's like, why did that happen? It's like, what a great question. I'll have to track that down. Yeah, question. Sorry, sir.
On the handout, it looks like the zygospore makes the spore.
Yeah, what's going to happen, this, it gets even more complicated, and so I usually stop at this step. But what happens is, okay, let me see if I hopefully I won't screw this up too badly. Okay, so during fertilization, the gametes are haploid, so they fuse and you end up with a diploid, zygospore, zygosporangium, and then they undergo meiosis, and that gives rise to haploid spores up there in the little cartoon. So, and you guys, I would never ask this on the exam, but what happens is, following meiosis, you'll get formation of a sporangium with genetically unique sporangiospores. They're haploid, but the genes in there are this recombination of genes from, we could say, mom and dad. Okay. On the lab practical, I never ask you about that next step where that new sporangium is formed, because it's just like, to me it's mind-boggling, and I've been teaching this for 10 years. So we usually just keep it right here, and we don't go beyond that. Now, if you guys do go into botany, plant biology, you'll go through the whole shoot and match, right? But for us, this is going to be enough. We're kind of looking at diagnostic stages, how you could diagnose the fungus that maybe is growing on a plant or animal, or growing in a plant or an animal. Okay, so thank you for bringing that up. And I'm sorry, you guys, I don't need to rob you of information. Okay, yeah. Okay, so let's just take a look at Penicillium. Okay. Now, it turns out that for reasons I don't understand, when your colleagues yesterday asked us, why did this happen? And it's like, oh my gosh, that's a great question. I'll need to track down on my colleagues and ask them. For reasons we don't totally, or at least I don't totally understand, some fungi have lost the ability to reproduce sexually. And all I can think of, there were mutations that knocked out genes that were important for sexual reproduction. That's kind of a weak answer. So I need to pursue that and find out what the mycologists think the reason is for a group of fungi known as anamorphs. Okay, so anamorphs, these are fungi who have lost the ability to reproduce, like its which have lost the ability to reproduce sexually. And the reason these guys are really important is, like, you know, gosh, way back in the 70s when I was going to college and we were studying fungi, the biology teacher always talked about this class called the Deuteromycetes. And these guys were nicknamed the Fungi Perfecti. They were considered imperfect because nobody could detect sexual reproduction. For a fungus to be perfect, it has to reproduce sexually, right? And these guys, nobody could find sexual reproduction. Okay, so with the development of DNA sequencing and RNA sequencing, they were able to do the nucleic acid sequencing on these guys and find out, oh, most of them genetically are related. We now know these guys belong to the class that we'll discuss called Ascomycetes. We'll come back and talk about Ascomycetes. But for some reason, they've lost the ability to reproduce sexually. And again, I need to find out, see, you know, what the mycologists think that's all about. If I just told you it's an advantage to reproduce sexually, why would it be a survival advantage for so many fungi to lose the ability to reproduce sexually? And one thing we were brainstorming yesterday, many of the pathogens are in this group. Okay, so many of them, the majority of the human pathogens are in this group. So there's many pathogens. And just in brainstorming yesterday, we were wondering, well, maybe if you've evolved this ability to infect and live within another organism, you know, that's a pretty steady environment. And just brainstorm, you guys, and just think, well, is it the pathogenic fungi that have given up sexual reproduction because the environment in their animal or human host is so consistent? Just brainstorming, you guys, and I'll try to find out. Because that was just such a good question, and I really didn't have the answer for it. Okay, so one example of an anamorph that you guys are going to be looking at today is Penicillium.